Welcome to Nikolay's genetics lessons, and today I'm going to teach you how to describe two-dimensional form of RNA using BPSEQ notation. In my previous videos I already described how to use dot bracket method, and today we are going to talk about BPSEQ notation, base pair sequence notation. And take a look at this very simple RNA structure. Every RNA starts with 5' end, as you see it is here, and ends with 3' ends, which is here. So this guanine here is going to be base number 1, and this uracil here, if you count all of them, is going to be position number 20 of the nucleotides in this chain of nucleotides. Because this is RNA, instead of, for example, thymine, we see here uracil. As you know, cytosine base pairs with guanine and uracil base pairs with adenine. But between cytosine and guanine, here is a triple bond. Between uracil and adenine, double bond. Hence, different colors between, as you see, adenine and uracil, it is blue, meaning double bond, and between cytosine and guanine, it is red color, specifying that here is triple bond. Otherwise, these colors doesn't mean anything for description of the three-dimensional structure of the RNA. So in order to describe this shape, so computers would understand we're using position, base, and pointer. So position of this guanine, we start with guanine, position number 1. We have here guanine. So take a look. I am drawing a table. So position number 1, guanine, and it points to other nucleotide where it makes a bond, which at the position 20, as you see. Next goes uracil. It is position number 2. Take a look. Position number 2, uracil, and it connects with position 19. Next go, as you see, at position number 3, cytosine, and it connects with position 18, or points to the nucleotide 18. We do not specify it yet, what kind of nucleotide it is. Next, position number 4, and at position number 4 we have uracil, and it connects with nucleotide 17. Next, at the position 5 we have guanine, and it connects with nucleotide 16. Next goes position number 6, where we can find adenine, and it connects or points to the nucleotide 15. Then goes position 7, where we have adenine, and it points to the position 14. And then we have position 8, cytosine, it points to the position 13. Then we have position 9, guanine, but it doesn't have any bond. So we put here 0. Then goes position number 10, adenine, and it also doesn't connect with anything. So here we have hairpin, and we put 0 here. At the position 11, adenine also doesn't point to any nucleotide. We put 0. And at the position 12, adenine also doesn't point to any nucleotide. We put 0. And here is interesting. At the position 13, we put guanine, and it connects to the position 8. So as you see, at the beginning we specified all the nucleotides here, but didn't specify any nucleotides here, only their position. As soon as after hairpin, we are doing opposite. We specify nucleotides and specify to which position they point. So at the position 14, we have uracil, which connects with nucleotide 7. And then at the position 15, we have uracil, which points to the position 6 and makes a bond. Then position 16, cytosine, points to the 5th nucleotide. And 17, we have guanine, which base pairs with position 4. And at the position 18, we have guanine, which connects with position 3. And 19, nucleotide 19, is adenine, which specifies, which connects to the position number 2. And lastly, we have nucleotide 20, which is uracil, which connects with position number 1. And we gave the full description of this RNA in a form which can be stored on the computers and in the form with which computers can work and would understand and can convert this table of numbers back into a picture like this. And this is all for today. Subscribe and see you in the next video. Goodbye.